Okay, so today we are going to talk about clustering. This is agenda for today. We will mention a couple of different clustering algorithms, and some of them are general ones, like k-means, db-scan, and spectrum clustering. We also will mention SC3, which is a clustering algorithm designed for single cell specifically. And they use a technique called ensemble clustering. And then at last, we will mention this hierarchy algorithm. Okay, as we mentioned from the overview of machine learning, so we have supervised learning, for example, regression or classification, and also unsupervised learning. Clustering is a task for a group of your data points or objects, whatever you call it, into the same group based on their similarity in some of the features. So this belongs to unsupervised clustering. So you don't have to teach or train your classifiers, not classifiers, clustering algorithm. You just give the input and let it to do it automatically. So it automatically organizing your data and understand the hidden structure in your data. If you have different clusters in your data, it won't be visible and using original data, but after clustering, you should be able to do. So in single cell study, we do this at the pre-processing step. You remember in my laptop MATLAB program, and last week, we talked about the dimensionality reduction. So you have a t-SNE embedding. The next step is from here to different color. Okay, so different colors indicate they've already been grouped into different groups. So that's the next step. So we are following this single cell sequence analysis flowchart, and step by step this week we move to clustering. So the purpose of that is after you cluster, you learn the features as a group for group of cells rather than unstructured data. So you can see average expression of certain genes for each group. If you show a difference, you know, okay, those are the marker genes for certain clusters. So that's our purpose here. And here is a real example. That's a real data set collected from my collaborators, one of them. And you can see this from a central nervous system, and you have a lot of astrocytes and the microglia and some unknown cells. And that's t-SNE embedding results like this. What do you think about this clustering problem? Is it easy one or hard one for this? Yeah, Alex.
It's probably going to be a little bit difficult just because of how spread out the astrocytes are.
Yeah, so for us, by human eyes, it's intuitively simple. You have 3 clusters, but it's difficult for algorithm because the density issue they're spread out for astrocytes there. So if you use general clustering algorithm without any no algorithm shouldn't be trained, but if you select the different algorithms, and then you get different results. So on the left, so those panels are k-means clustering algorithm gave you results, and some right side are different algorithms gave you slightly different results, and those differences are color coded. So you see this is the right solution which fit our understanding of data. So that's where our expected results. But remember, we are visualizing 3 or 2 dimension, and if up to higher dimension, then clustering algorithm may rely on higher dimension classification and may give you slightly different results. So that's also expected. Okay. So major clustering approach, we have partitioning, have hierarchy, and model-based, density-based, graph theory-based. There are different ways to group classification algorithms. Not classification, I always get confused with clustering approach, not classification. They're quite different, okay? So we have major clustering for clustering approaches. Well, the most general purpose clustering algorithm is k-means, and it is a partitioning approach. And for model-based, like Gaussian mixture model, it's another way you model your data using Gaussian distribution. And the DB scan is a density-based. Another density cluster, which is relatively new publication, propose this, is also density-based. We also have so-called affinity propagation or spectrum clustering. We are not going to talk about affinity propagation in this class, but the reference is there, and it's also a really good algorithm, and you can change the parameters and ask for different size of those clusters. Pretty awesome. So today we are going to talk some of them, but not all. Okay, let me see. We are going to switch to k-means algorithm. And you see this awesome equation. So I'm going to explain this on Blackboard. So this intuitively sound, it's very simple. And after the class, you will think you may be able to design a clustering algorithm yourself if you know those basic operations. The most important one is to measure the distance between points. Okay, so I'm going to switch my gear to before my laptop died off from it. Okay, we need define the data points. Okay, you know we use vectors. We use vectors. Here we have A. We have a random number here, 1.5. $1.5, 2.5. That's my A, right? So vector we represent with this vector plot here. And see, okay, I'm going to randomly put a point, say B, okay, in this case, 2, 1. Okay, so right here, B. So to compute the distance between these two data points, it's very easy. You can see A minus B. Okay, so A minus B is another vector, which if you compute this out, one minus B minus 5 plus 1.5. Okay, so that's our new vector. New vector is, so if that's 1, so minus 1.5 here. 0.5, 1.5. Let me change the color. So this is my the difference between these two. We have a new vector. Okay, so this is A, B. Right? So if you can see from here, if you draw a vector, that's also the difference between that. But if you start from the origin, then that's A and B, A minus B, right? In order to compute the distance, which we are going to compute the length of this. So what's the length of this vector? Is the norm of it. Right? The norm. So norm is okay. So A and B is 1.5, 1.5. Okay. The norm will be 0.5 squared plus 1.5 squared, right? Okay, so that's the distance between A and B. So for any given point, if you have their coordinates, just do that. Now, if I have a lot of A on the plan here, so I have another A. So this is A1, A2 here, 3 here, 4 here, 5. It doesn't matter. I have a lot of A. And B is my random number. So I select the B. Then I can compute the distance from a B to all of them. Right? So I will compute A1 minus B. Okay. A2 minus B. A3 minus B. And then I can sum them together, add them together. Right here are the sum of distance from all AI. So we have a lot of A, I equals N, so depending on how many. So I do 1 to N, depending on how many points you have. And the B is one random number select. So to make this simple, we can rewrite this into a sum of AI minus B. So I is 1 to N. Okay. So because we know how to compute for one of the point to B, so A1 to B, A2 to B, and put in the norm formation and make a sum. So what's this sum? So that's the overall distance from all the points to select the B. So you can move B to somewhere else, and then you compute this. Right? You can move B, but that's very easy computation. All the points minus B and take a norm and sum them. All right. So that's the equation on on your slide. I cannot show you a slide here. Okay. Now, we know this equation, we are going to do k-means clustering. The example I have is something like this. I have some data points. Doesn't matter. There's something like this. Okay. So for k-means algorithm, I will select number of k. So you have some idea how many k's on your system, and you don't have to know this. And there's because this computation is so fast, so you can see I want to try k equals 2, or 2, 3, 4, 5, and then there's a way to judge so which one is best. Okay, but for this example, we assume k is 2. Okay, once you have k equals 2, you have to say, okay, I'm going to generate 2 random numbers on this space. So what I'm going to do is to randomly generate 2 numbers on this space. One I will put here. Another I will put here. Okay. It doesn't matter. You have to generate 2 random numbers. I will assume this will be the central node of 2 clusters, because I'm going to make 2 clusters. I will think, okay, around this point, random number, there's 1 cluster here, and for blue one, I have another cluster here. Okay, but certainly this is not a good separation because we haven't done anything computation yet. We are going to iterate this. The first step, you are going to compute the distance for each number. You know how to compute the distance from all the white points to red, okay, using the equation. All the A, I minus, if you think this is B, B1. Remember this, so you can compute. You don't, right now you don't need the sum. You can compute each distance to here, and you also compute the distance point to blue points. Okay, if this distance is close to red one, and you think, okay, this will be number of that, and this one, and it maybe belongs to blue. So it's still red. So this like attractor, so you attract all this to right. Okay, you have to, in the computer, you have to define a way to group them into 2 clusters, put a label. So this label will be 1, that label will be 2. In this case, you already have a separation in the cluster. But your separation is something like this, right? Separated by distance. We haven't done any iteration yet, so next step, we will compute the central node for the right group. So this is the right group. We will take average about all their x-axis and y-axis. Somewhere we will move our central node to here. So that will be the cloud, right cloud center. And for this side, the blue point, the center looks like somewhere here. Okay, so I'm going to move my central node from the original random guess to some educated position. That's my first step. So I move my original random assignment of the centroid from red original position to this one, and move the blue one to here. Now I'm going to start again and remove all this membership IDs and recompute the distance from all the points to, okay, all the points, right one. The new, okay. I don't know, this is close to that one still. Okay, after this step, we eventually introduce another last string separation with the two groups. And we will redo this, iterate this for a couple of times until there's some criteria that we can stop. Okay, in this case, it's very easy. After two iterations, maybe the membership of your data point will not change anymore. We just compare this step, this next step. If no change, we stop the algorithm. But that's only two-dimensional space and the simple examples. For more complex situation, you may still have to struggle to see, okay, my membership is still changed when they update this. But how can we stop? You can stop, see, I want to have a hard cutoff after 100 iterations. I will stop my algorithm. I will satisfy the solution whatever the algorithm gave to me. You can also see, I only try 50 because I want to save time. You can try different starting points randomly. So this time I start random points from here, and give me the solution like that. I will see, I try another initial values after 50 iterations, whether I still have similar results. I can combine this. So there are different solutions of the principle is so simple, right? You just need to know how to compute the distance between points. Okay. Any questions? Yeah, Alex.
So how does it decide what the initial cluster or the initial iteration?
The initial, you don't have to decide. You have a random number, you just put two, I just close my eye, put 2 points.
Yeah, the initial separation to determine, or the initial labeling to determine what belongs in group 1 versus group 2.
There's no initial labeling. I just put a random number here and then see the distance between them. If it's close to red one, it belongs to red group. Close to blue one, it belongs to blue.
And it, does that labeling get iterated on further, or with successive iteration?
Yeah, the labeling will not carry along. The labeling will only see, okay, the blue one, I'm going to compute the central node by average. So the first step, remember, you have a blue cluster here, right, and we compute the average blue point, X, Y, and then find the new center of the blue cloud.
Okay, so it just picks the 2 points, does the labeling, then and then calculates the distance. Takes 2 new random points, does more labeling, and then the distance again.
Yeah, so once you compute the central node from your cluster, and then you forget about all the labeling and use these 2 points as a new central node and recompute the membership. And then use the membership, recompute their central. Yeah.
Does it take in like the blue group? Does it measure the distance between like the red group? How does it stay on the right side?
So the first step you
okay, you separated them.
You separate them, and you know, okay, the point close to the blue points are blue group. So you will use that into the computation of the next central node. Yeah.
Can your cutoff point change with successive iterations? Or once the cutoff point is set, does it stay there to separate the two groups?
The cutoff, this cutoff, the line I draw, will not be in the algorithm. I just tell you
It's
there
just
for illustration purpose. Yeah, we don't have any expression of this. Yeah, but it's just a number sheet, depends on the distance between points.
that makes more sense.
Yeah. This algorithm is so efficient computationally, so you can use it widely whenever you need it. You can define the distance. We know there are different ways to define distance, right? So you can plug in your distance definition. Not only, this is the easy one with Euclidean distance, but you can define your own distance, you define your own similarity between data points, and plug in this algorithm. You can learn the features using dimension reduction to reduce the number of features instead of use OGN's expression to define the distance between cells, right? So there's different ways to customize the details of this algorithm. Okay, on the screen, that's exactly what you see here. The U is average at the central point of your cluster, and X are the points belongs to same group. So you want to make this small as possible, the sum of the distance from the members of your data points to the center as close as possible. Make this, you select the central point, the central node point, make the cluster, this sum of distance smaller. Okay, so this is similar as a minimization or optimization problem. So you want to minimize the sum of this for their perspective centers, because you have multiple centers. You have k number of centers. We want to compute the center for the right point and compute this. You want to compute blue point. You have same expression for blue point. And make this small, this small. That means they all shrink into their own number cloud. Okay. So the way I just showed you to iterate, just guarantee you will find the centers for your clusters, and then eventually make this the sum of these two become smallest. So I have to express this and times 2. So that's the sum for the members up here. That's members here. The total of this, the whole thing should be minimized. Okay, you just cannot make this small, but make this big, or the other way. So you have to balance between different clusters, make the total of the distance to their central node smaller, as small as possible. That's situation K2. If you have 3, then you have another term. Okay. Okay, the equation you see on the screen here. The first is every individual cluster. So on this plot is this k equals to 2. I have a ki, so ki, one is this red point, another is this magenta, the pink one. And then that's the total for each of them. Then you sum that again. So that's the expression here. Okay, they use squared, same. Squared no. Okay, so that's the text description of the algorithm. So just like a story. So you decide on the value of k first and initialize the k cluster centers. That's a random number we generate on this two-dimensional space. But if it's high dimension, you have to generate a random number of high dimension. Depends on the dimensionality. And then decide the class membership by compute the distance between your data points you want to cluster to be clustered and into those random center. You just randomly generate it and estimate the center again, and then move and repeat. So that's iteration. Okay, so those expressions are what I explained on here. Yeah.
So just make sure, so when it's re-estimating the centers, that's when it's trying to find the center, right?
Yeah.
Right, okay.
So random and then group by distance. And using the yellow one to recompute the center. Okay. And recompute the member. And then eventually recompute the membership, and eventually you stop there.
So the membership is essentially like in a
The distance.
yeah, when we're assigning names to our clusters, our cutoff line is essentially the genes that identify each cell type and like how they're expressed within like neutrophils versus the
Yes, so yes, for single cell study here, we haven't yet talked about the cell type. So we don't know the cell type. We did T-SNE embedding based on their distance on high dimensional space. You reduce into 2 dimension. So you see them when they close by, they maybe belong to the same cluster, same cell type. But we don't know them. But in the correct resolution, you should have same cell types like B cell, T cell, and macrophages should be grouped together. And that grouping may be not so obvious, just like the example here. Then we use clustering algorithm to help us to make them into small clusters. But you'll still be able to separate B cells into 2 groups as long as you have 2 subclusters in there. The algorithm don't know anything about those. They just look at the coordinates, right? So the input for this algorithm is only their x, y coordinates. Nothing about the other genes expression because we already did the dimensionality reduction. So now the input for k-means algorithm clustering is only the position. Yeah.
And when are we trying to minimize that distance again? Is that just another way to calculate the center in a way?
Yeah, so this procedure guarantee you at the end you get this.
Right.
But if you don't want to use this, their general purpose optimization algorithm, you see, I want to find k numbers of central node. And the algorithm will use this loss function to, or penalty function or objective function, whatever you call it, will help you to find the central node. And the solution will be the same as what you did here.
Right, yeah, it's just instead of trying to calculate the mean centroid, you just try to minimize the distance.
Yeah.
Okay.
That's right, yeah. So the general objective is like this, but the procedure is not to look at this per se, but will guarantee you get the same solution. Okay, we move to the next one, Gaussian mixed model. And remember, we talked about if you know the distribution of the human height, then you have model, okay, Italian have one, the shape will be the same, but the mean slightly different. But here we just assume our data follow those Gaussian distribution. And if you know the cluster, like this blue points and the yellow points, this example is one dimension, and you can fit like two Gaussian curve, okay, to say, okay, I have a mean, I have a standard deviation. So you know how to fit this to make the likelihood to explain those points collectively maximized. But in clustering, we don't know the membership. So in this case, we have no idea where the blue point or yellow point, the membership are, but we just look at the distribution and see, okay, if I use two Gaussian curve and fit this, and I explain my data very well, and I will see, okay, under this curve, and those are the blue points, and these are the yellow point. Of course, there's mistake because we don't know this yellow point is such an outlier and into the blue point region, but because we fit this model with this roughly and gave this. Okay, this is just one dimension. If you have two dimension, you have a space, surface, a high dimension, do the same thing. But you have to assume your data, each variable follow normal distribution in order to use this. But statisticians, you like to use this because Gaussian has been such a well-studied model, and there are a lot of nice properties make this very easy to compute. Or there are a lot of theorem you can use, you can borrow. Still very powerful way to do it. Well, k-means and also this Gaussian mixed model also have one weakness, is that they're not applied to this so-called non-convex shape. On your left side, there are cloud. If your data point follow that within that shape of the cloud, it's called convex. And the non-convex, you have this this non-convex shape, which I don't know how to explain this mathematically. But when you say it, you know this shape is non-convex. Okay, so that's some examples. If you have a real separation of the membership of two clusters on your left side, this column, and when you apply k-means, you can separate nicely for those two clusters, but you cannot apply to this non-convex shape when data points make this kind of cloud, non-convex shape. In that case, you will select the central node here for red group and a blue group. On the right side of this panel, also you see two columns of this, and also this is the correct expected separation. You see this complex shape, and the k-means just no way can find a solution for that, okay? And also because you have the non-convex shape of the data cloud, k-means will have two convex shape separation into clusters, so you just cannot do it. So that's where the DB scan algorithm comes in. And if you look at the paper published in 1996, so this is a relatively new algorithm. I don't know, to me, and 1996, just a couple of days ago. So it's a relatively new algorithm. But if you look at the simplicity of the idea, it's nothing. Okay, you follow the neighbors by, okay, it's close by, it's your neighbor, you keep it in the group. If it's not, you're not a group. That's all the idea. So you see such a commonly used algorithm was only invented in 1996. So you have a chance, if you have a good idea, you just tweak some your algorithm and borrow something else, you may make a new algorithm very easily. Okay. So what's the solution for this kind of shape? What do you think? So idea is just follow small steps. If you have a neighborhood, you include them and slowly grows. You don't jump, then you will be able to recover this, right? You don't jump to a long distance and found the membership. That's exactly this algorithm is doing. I don't think this is a very elegant algorithm. You have to define two parameters. So how large your radius is. So if you are using so you have to define two parameters. One of this is what's the neighborhood, the size of neighborhood, okay? And then you have to define the minimum number of points in this neighborhood in order to become a new cluster center. So you have to have enough members. So once you have these two, you just gradually expand your neighborhood until you cannot. And then you define new cluster center, expand. At the end, you get the shape. So let's see, I'm going to play two videos, explain almost the same thing.
I need to find the best way to learn math and science.
Hello and welcome. In this video, we'll be covering dbScan, a density-based clustering algorithm, which is appropriate to use when examining spatial data. So let's get started. Most of the traditional clustering techniques, such as k-means, hierarchical, and fuzzy clustering, can be used to group data in an unsupervised way. However, when applied to tasks with arbitrary shaped clusters or clusters within clusters, traditional techniques might not be able to achieve good results. That is, elements in the same cluster might not share enough similarity, or the performance may be poor. Additionally, while partitioning-based algorithms, such as k-means, may be easy to understand and implement in practice, the algorithm has no notion of outliers. That is, all points are assigned to a cluster, even if they do not belong in any. In the domain of anomaly detection, this causes problems, as anomalous points will be assigned to the same cluster as normal data points. The anomalous points pull the cluster centroid towards them, making it harder to classify them as anomalous points. In contrast, density-based clustering locates regions of high density that are separated from one another by regions of low density. Density in this context is defined as the number of points within a specified radius. A specific and very popular type of density-based clustering is dbScan. dbScan is particularly effective for tasks like class identification on a spatial context. The wonderful attribute of the dbScan algorithm is that it can find out any arbitrary shaped cluster without getting affected by noise. For example, this map shows the location of weather stations in Canada. dbScan can be used here to find a group of stations which show the same weather condition. As you can see, it not only finds different arbitrary shaped clusters, it can find the denser part of data-centered samples by ignoring less dense areas or noises. Now, let's look at this clustering algorithm to see how it works. dbScan stands for density-based spatial clustering of applications with noise. This technique is one of the most common clustering algorithms, which works based on density of object. dbScan works on the idea that if a particular point belongs to a cluster, it should be near to lots of other points in that cluster. It works based on two parameters, radius and minimum points. R determines a specified radius that, if it includes enough points within it, we call it a dense area. M determines the minimum number of data points we want in a neighborhood to define a cluster. Let's define radius as 2 units. For the sake of simplicity, assume it as radius of 2 centimeters around the point of interest. Also, let's set the minimum point, or M, to be 6 points, including the point of interest. To see how dbScan works, we have to determine the type of points. Each point in our data set can be either a core, border, or outlier point. Don't worry, I'll explain what these points are in a moment. But the whole idea behind the dbScan algorithm is to visit each point and find its type first. Then we group points as clusters based on their types. Let's pick a point randomly. First, we check to see whether it's a core data point. So, what is a core point? A data point is a core point if, within our neighborhood of the point, there are at least m points. For example, as there are 6 points in the 2 centimeter neighbor of the red point, we mark this point as a core point. Okay, what happens if it's not a core point? Let's look at another point. Is this point a core point? No. As you can see, there are only 5 points in this neighborhood, including the yellow point. So what kind of point is this one? In fact, it is a border point. What is a border point? A data point is a border point if A, its neighborhood contains less than m data points, or B, it is reachable from some core point. Here, reachability means it is within our distance from a core point. It means that even though the yellow point is within the 2 centimeter neighborhood of the red point, it is not by itself a core point because it does not have at least 6 points in its neighborhood. We continue with the next point. As you can see, it is also a core point. And all points around it, which are not core points, are border points. Next core point, and next core point. Let's pick this point. You can see it is not a core point, nor is it a border point. So we label it as an outlier. What is an outlier? An outlier is a point that is not a core point and also is not close enough to be reachable from a core point. We continue and visit all the points in the data set and label them as either core, border, or outlier. The next step is to connect core points that are neighbors and put them in the same cluster. So a cluster is formed as at least one core point plus all reachable core points plus all their borders. It simply shapes all the clusters and finds outliers as well. Let's review this one more time to see why dbScan is cool. dbScan can find arbitrarily shaped clusters. It can even find a cluster completely surrounded by a different cluster. dbScan has a notion of noise and is robust to outliers. On top of that, dbScan makes it very practical for use in many real-world problems because it does not require one to specify the number of clusters, such as k in k-means. This concludes this video. Thanks for watching.
I have one question about that.
Yeah.
It said your n has to equal 6, but on the right cluster there was only 5, yet they still define that as a 4 point.
Oh, I didn't see whether they're cut the boundary. I don't know whether.
I had the same question.
I thought it was about the same for the bottom.
And then see the bottom one too.
Oh, really?
Yeah.
I have to review that myself again.
I mean, the concept makes sense. It's just why they put those.
Okay, okay. So maybe it's a mistake in their demonstration or something. Okay, I have to go back and take a careful look at this. So next time I may not use this, but we have another video to explain. So if you want to try, see whether this makes sense, but they're explain the same idea. So to me, this is just a simple idea, and you just follow and like a worm to climb to a different branch of the tree. I mean, it's really nothing. Okay.
The explanation was really good.
Yeah.
It made sense.
Yeah. So the limitation is you have to define these two parameters. If you define them differently, you get quite different results. So in single cell study, we don't use it because I don't know how to define the radius and define the members in there. And if I change the parameter a little bit, I get quite different results. That's not something I really want. So yeah. That's a limitation. Let me see my battery. Oh, I still have enough time to explain to you two concepts before we make a break. Okay, I want to explain Laplacian matrix because we are going to use it in spectrum clustering next hour. So I will explain this, then we take a break and we come back. The online student have to log in from a different the Zoom because I'm going to shut down this computer and use another. Okay, so Laplacian matrix is a difference between two other matrices. So L is the difference between D and A. Okay, when you if one matrix minus another, the shape should be the same, right? So the D and the A are the same shape. Actually, they all square the matrix. If you have n nodes, so it's n by n, A also n by n. Okay, so that's the dimensionality, and L must also be by. So what is A and D? So A is called adjacency matrix. A is this describes a connectivity between networks nodes, and the D is degree matrix. Okay, so once we have these two matrices, we can easily compute that, and that's our foundation to do the spectrum clustering. So this adjacent matrix is for network stuff. So if you have, for example, have a network like this, so we have 1, 2, 3, 3 nodes, and 5, 4, 6. Okay, so then the adjacent matrix will have 6 members. So 1, 2, 3, 4, 5, 6. That will be 6 by 6. Not going to complete it because it's so simple. And if this network, you can think about those as a group of cells. You have 6 cells, and your distance between cell 1 and 2 is short enough to allow them to be linked. Okay, so you have some link like this, and you can set up a cutoff. The distance between 2 cells has to be small enough here to make them link. And also, okay, if by chance you have something like this, you have a graph. So you build a graph between all the cells. This graph is not fully connected. So for example, 2 is not connected with 6 directly. So 2 to 6 will be 0. 6 to 2 will be 0. If they are connected, you put 1 here. So 1, 2 is connected, 1. 1, 3 is connected, 1. 2, 3, 2, 3 connected. And for themselves, they're 0. So you fill this up, and then you get adjacent matrix. So this network can be represented by adjacent matrix. And this is symmetric because there's no direction between nodes. Okay, so I can fill this in according to my okay. Easy, right? So once you have this, you are going to compute the degree matrix. That's even simpler, and also you have a 6 by 6 matrix. Use this index. The degree matrix only have values in their diagonal lines here, so the rest of them are all zeros. So what's the value here? For 1, you will see, okay, how many neighbors it has. Okay, in this case, it's 3. That means the degree, the connections are linkers of the node 1 to others. There are 3 possible links to other members. And for 2 is 2. For 3, 1, 2, 3, 3 linkers, and 3. So you just fill this out. Yeah, Alex?
So that's just pretty much the sum of a row or a column.
Yeah, exactly. So if you have adjacency matrix, you can compute D by take the column sum and put in the diagonal line. You're right. So you don't need a degree matrix because your adjacency matrix will automatically help you to generate a D matrix. And then you take the difference between D minus A. That's your Laplacian matrix. Remember, this value can be weighted. See, I want to have 0.5 weight, and here is 1.5. This will be 2. This is 0.6. It's possible. In that case, instead of putting this at the 1 and 0, you put their weight into adjacent matrix. It become a weighted network. You can do the same thing. You just sum the column and then you get degree. So in this case, could be 35.3 at the end. Somehow you have a degree matrix, which again the sum of this. Okay. Yeah.
I'm sorry, the weighting is the distance between points as opposed to either.
It doesn't have to be the distance. It could be some other properties. Yeah. So you, yeah. Yeah, so in this case, if you use distance as your cutoff, so this case, the link between them, yeah, it could be the distance or the opposite of distance, whatever. There are different ways to compute the weighted network. So once you have this, you should have adjacent matrix and then the degree matrix. Okay, so we can stop here. So you know how to compute the Laplacian matrix. Take no effort, pretty easy, right? As long as you know how to compute the distance between cells. You know how to compute the 2 point, the distance. And you know how to make a huge network if you have a lot of cells. And then you know how to make an adjacent matrix. That's n by n. Depends on how many cells you have. Yeah.
Is there a reason why they use subtraction instead of addition? Because the diagonal row for the adjacency matrix has no value, and that's where the information in the degree matrix is stored.
Yeah, yeah. So at the end, this value, this something like this. You have all the values 3, 2, whatever 3 in this, and along -1, -1. But, yeah, because that's 0 minus indicator 1 here. So you have something like this. So we are going to use that.
Sum it up, it all equals 0. So it's like a checksum for them.
You can check this. The sum will be 0. Yes, column sum. I think so. So this matrix contains the information. But yeah. So. Okay. It's time to take 5 minutes break, and I will switch my computer, and online students, please log in again, okay? Testing. Online students, please let me know.
We can hear you.
Okay, good. Okay, thank you. So we mentioned the limitations for this dbScan algorithm is you have to put 2 parameters, that 2 parameters has to yield the results. So in order to select the correct numbers of these parameters, there's a new algorithm, just like a extension of dbScan idea, and this algorithm published in 2014. Okay, so you see, if you can see here, I'm not going to discuss how these parameters will be defined and determined by this algorithm. That's your homework. You are going to find this paper published in Science with the title and read through and see, okay, what technique they used to determine number of the clusters. For example, you have input like this. To me, it's clear, and you have those clusters, but it's not so easy if you don't see if they're in high dimension. If you the algorithm will not be able to guess what parameter it will use. But this algorithm, okay, using a technique to find the number of this and then give you the correct classification. the technique they use is two parameters of this to separate data points or cluster centers and found outliers. So you have to read through what's the local density and say, okay, it's here the distance, something like those, and then try to understand. So no any help, and just read the original paper to understand the machine learning paper published in Science in 2014. So this is a good training for you, I think. So that will be the homework. Okay, so next, spectrum classification, clustering, not classification, again, sorry. You already know what is graph Laplacian matrix. The rest of them should be very easy. So this is an algorithm based on graph theory. And in graph theory, we care about the groups or modules or subnetworks or members, whatever you call it. So like in this human-human interaction network, you have family members as one subgroup, you have a school friends subgroup. So we want to learn from this social network, what's the relationship between members and whether these members in what different membership subgroups. So we really need those algorithms to define modules. Modules also called cluster, okay, in terms of networks. And these clusters or modules are characterized by the dense connections between members within the cluster with this weaker links cross modules. So you try to identify those kind of things. It's a big topic, right? Facebook want to know how to do this, maybe. So if you do this based on this graph theory, spectrum clustering, you can apply to different kind of data shape. It's okay to do for this, classify them to 3 groups. It works for this kind of separation. It works for that. So it's pretty powerful, right? That's exactly on the whiteboard I just showed you. This is unweighted network. That's why you put just 1 as indicators of they are connected. That assumes the weight between those linkers or edges are all 1. Then you can put different weight in there. Then the degree matrix, just like Alex discovered, then you can just sum the column. So the total of this column will be 3. So 3 will be, sum this will be 2. Sum this will be 3. So that will be put into the diagonal position in this degree matrix. The other positions are zeros. The difference. Okay. So what's next? So for spectrum clustering, next step is to do eigen decomposition. We come back to the eigen decomposition again. So so this Laplacian matrix contains information from original network and also processed. So you have those information in the diagonal position. And once you found its eigenvectors, that's its eigenvectors. So what's eigenvectors? So if you have this Laplacian matrix, it's L, and you times its eigenvectors L times X is lambda, which eigenvalues times X. So that's a definition for eigenvalue and eigenvector for L. So we can move this around, move this X to here. So you can rewrite this as L to be decomposed into X in -1. This is a, and X, right? So this X -1 means inversion of that matrix times its, so inversion of this eigenvalue matrix times lambda, which is eigenvalue, and that's eigenvector matrix. So you decompose that, and the information we will use is inside this X matrix. This is eigenvector matrix. We are going to use the second smallest. So we sort the columns of eigenvector matrix by the eigenvalues and reorder them. And we use the second smallest eigenvalues corresponding eigenvectors as our new variables to separate the graph. So if you pick that out, so because this is the second smallest eigenvalue, and that's the corresponding eigenvector, we pick them out, and we to do the clustering using this as a new variable. Okay, use this as a new variable. You separate nodes by this value. Okay. So here we may use k-means or whatever algorithm because you define those nodes 1 to 6 using a new collection of variables. These variables will compute out using eigenvectors from a Laplacian matrix. This contains the information tell you how to separate graph into two. And you can use more. So in this example, we only use just one smallest. You can use 3 smallest eigenvectors to define like a 3-dimensional and then classify. This whole procedure, it's very easy because you know how to compute the L matrix, you know their built-in function, you can compute eigenvalue and the eigenvectors, and then you pick smallest eigenvalues and to do clustering using this to classify, to do clustering to cluster points in this graph. This separation is between 1, 2, 3 will be one group, and 4, 5, 6 will be another, depending on these values. So some positive, some negative. The whole procedure can be applied to weighted matrix, weighted network as well. So the same 6 nodes. In this case, we have weight for each linker. And its adjacency matrix here, instead of A, we use W because it's weighted. You have those values between node 1 and 2, 0.8, because on the graph is 0.8, right? Between 1 and 5, 0.1. So between 1 and 5, 0.1 here. And same thing for degree matrix will be the sum of the column here. And you take total that, you put in here, and all other places are zeros. And then the difference between this is Laplacian matrix. Okay, you can try this, put in this matrix in MATLAB. And then here you get your eigenvalues. And this is the second smallest. And the first one, there's no information for the first one. First one, the smallest eigenvalue corresponding eigenvector is this. No information because they're identical. There's no information to help you to separate the. The second one give you the information. The first three are positive, and the last three are negative. So you can use this to separate them. If not a resolution not enough, you can use another smallest. So you combine these two to help you to separate. In this case, you have a two-dimensional description of your points. Okay, you convert your network stuff position on network and come back to these eigenvalues. The values in the eigenvectors. Okay. Okay, so this illustration is for using the second smallest eigenvectors to separate the points. Okay, we apply k-means onto a graph data. This graph data has been converted using Laplacian matrix, using eigen decomposition, using eigenvectors to do the k-means. So that's called the spectrum clustering. I believe if you try to understand all the theory behind it, how people invented, I don't know how they figured it out. But looks like the smallest eigenvalues contains information about the whole graph. And the second is the top cut the graph into two, and the third maybe cut the graph into three. So you have this spectrum, means you have a different frequency of different levels of cut and put together. So that the information is embedded into this eigenvector matrix. All right, there are some links to the reference to the YouTube explanations for this. I hope you can get enough information of that. So in Thursday, we will try to load example data. So this data is the one I just showed you, the real data in action. We use k-means to separate these coordinates. So there's no information about expression level whatsoever. This example data only have one variable s. S have two columns to put the position of these points in this two-dimensional space. That's all the information for clustering algorithm. So we lost a lot of information we threw away and only use the summarized information that's between cells relationship and their position. K-means will give you separation of this, that. Okay. Spectrum clustering is also a built-in function in MATLAB. Gave the same input S with position of the cell and ask for 3 clusters, it gave you this. Okay, we should have a DB scan. We can search whether we have DB scan in MATLAB. Then in that case, we need a parameter, 2 parameters, right? And also we cannot ask how many K we will generate it from the DB scan. Okay, those are the synthetic data, and we know the membership supposed to be, and that's the method. Here is clustering, spectrum clustering. That here the DB scan we mentioned. The other we mentioned the affinity. So you see the outcomes of this unsupervised clustering algorithm, and the spectrum clustering looks pretty cool, actually. Okay, so here are some blog articles about it. Apply k-means and DB scan and another cluster algorithm to the same data and try to compare what's the difference between them. You can try them out. And there are also research papers just compare and evaluate different algorithms when they apply to single cell RNA-seq data, right? And as you can see, the parameters matters, matter, and also the algorithm, the choice of algorithm matters. So now the idea is to try to combine different combinations of parameter settings, and also maybe combine different algorithm and compare the outcomes. If, for example, two cells, no matter you use whatever algorithm, whatever combinations of the parameters, always be clustered as the same member, same clusters, then these two cells may be really close to each other. So you can confidently see, okay, these two should be the same group. So that's the idea. You want to collect all the information you can have in a reasonable time to compute and then combine the results. Okay. Well, how to do this is also a subject of the research. So I'm not going to go that deeper. But there's the next algorithm, which people have been found that in single cell world, it's one of the best algorithm, uses this idea. So we can take a look of how this algorithm was designed. Then we know this called ensemble idea about those. So this algorithm called SC3, I forgot when it was published. It published in Nature Method. The idea here is to use different parameters to do a lot of k-means clustering and then combine two cells, always combine and to be determined as the same group, same cluster. Then you have a high probability to put them together. So that's the last step. You do a lot of k-means and you get a lot of results and combined. So how the parameters combinations have been set. This algorithm, we start from the matrix, the big X matrix. Remember, we have n numbers of cells, p numbers of genes, and then we do a filter, and the sum of the gene just have too many zeros, not will be very useful to do the clustering. So we filter them out and discard and only use mostly informative genes for clustering purpose. And once we have this clean matrix of gene and the cell, and then we will compute a distance matrix. They compute 3 different matrices. One use Euclidean distance. So the distance is between cell to cell, cell to cell. So if you have n cells, so this matrix, distance matrix will be n by n. The first one is easy, so Euclidean distance, just like we compute the distance between two vectors, you just consider the high dimensional vector. And then they compute the correlations. Okay, then you do the correlation analysis. So cross the two cells, cross all the space of the genes. For gene A, if this cell express high, another cell also express high at the same time, so you have a strong correlation. Again, Spearman correlation is also correlation analysis, but using non-parameter. So you don't have to assume some kind of normal distribution. So it's just by rank. Then if two cells has strong correlation and the distance between them will be small. So you have 1 minus R, which Pearson correlation coefficient, then you get the distance matrix. Whatever, so you have 3 distance matrix computing. Then we are doing the 2 transformations for each of them. For Euclidean distance matrix, you do PCA transformation, so you get the PCA matrix here. And you do Laplacian matrix, Laplacian transformation, just like what we did there, to get adjacent matrix, because the distance can be used as adjacent matrix. And then compute degree matrix, and difference between them, their Laplacian transformation, right? And the PCA is also some kind of summary. Okay, for each of these distance matrices, we do these 2 different transformations. So now we have 6 matrices. Okay, so you can see, okay, from the original data matrix already compute the distance transformation. So we summarize the information into 6 different matrices. And then that's not enough. For each matrix here, we will slide this by different range of D. So you use all the information in that matrix, or use only top D. So you increase the numbers of the information you include inside this matrix. Each time you, so this D range apply to each of the 6 matrices. But each time you compute a k-means algorithm to compute the number, making them into clusters. And then you have this results of the k-means algorithm applied to different distance matrix, transformed differently, and use different range of the parameters inside this transformed distance matrix, and to do the clustering. So once you finish this, you may end up with several hundreds of them, and then you combine to compute a consensus. That's the outcome. Okay. Can you tell me why this algorithm is not widely used so far? That's right, yeah.
So the consensus matrix, that's all of the data of all of the 6 matrices that have been transformed, where they all agree.
I don't know exactly inside there how they vote for this. Maybe just take average. We have to check the algorithm. I re-implemented the whole thing, but I forgot exactly how this consensus was made. Again, as this, how can you compute the consensus? Also, there are different ways to do it. But I want to focus on this top ones. Okay, so the clustering results of this will be slightly different from you use only k-means applied directly to the coordinates of the cells, right? We will try this in computer lab and use SC3 to cluster our cells, and then you do a different color to show them in the 2D or 3D embeddings, and you will see some of cells, although visually it's has been classified in this group, but it's colored differently. So that's inconsistent between them. I think they published in R, and we have a MATLAB version. Okay, so we are going to end, just quickly mention this hierarchy algorithm. This is based on the trees. So before those ones, the DB scan, k-means, or spectrum clustering, they are partitional. We don't consider as a tree structure between but a hierarchy characters here is a trace and relationship between them. Then at one of them to make clusters, and under the same branch cluster. Well, we use this very widely in the tree building algorithm that has been studied in molecular evolution. In the phylogeny, you're trying to build a gene tree or species tree. Well, we just borrow those algorithms, for example, dendrogram, and then you just plot them by distance and the membership, and you can see in this example, you have this tree with two long branches. That's some kind of a distance between to summarize the group of this. So you can see I want to cut the tree into two by looking at this longest internal branch, and it automatically cluster your cells into two groups. And then within this sub branch, you can cut from here, for example, then you group at a different levels. So there, and then they said this can be used to identify the outliers. Okay, you have this outlier, which has no members in there, only itself, so you know it is. And we do have some software package and implement those algorithms. For example, the Dendra split. I don't know when it's published. I never heard about the other people use it later. It's just a method here. Again, you process the matrix and build a distance matrix. Many this phylogeny or molecular tree builder algorithm is start from a distance matrix. So once we have distance matrix, there are many algorithms you can select to build the tree. Another algorithm is called the PCA reduce. This is published in 2016 in BMC bioinformatics. So that's another example. People use hierarchy clustering to do this algorithm, to do this clustering. Some advantages here, and I believe the the problem is it's not scaled up very well. So once you have many cells, you will at least you need to have a tree to describe this relationship about if you have several thousands of cells. Even to describe this tree structure will be an issue. Okay, that's all the slides I want to go through today. Looks like we end slightly early today, but I certainly can take any questions if you have. I will play the second video for DB-scan and see whether we will have something.
We'll be covering density-based clustering. Density-based clustering is also known as DB-scan or density-based spatial clustering of applications with noise. It works by defining a cluster as the maximal set of density points. There are two parameters that are taken into account. Minimum point, maximum radius of the neighborhood, and minimum points is the minimum number of points in the epsilon neighborhood to define a cluster. There are three classifications of points. They are core, order, and outlier. A core point has at least minimum points within its epsilon neighborhood, including itself. These are points that are at the interior of the cluster. A border point has less than minimum points within its epsilon neighborhood, but can be reached by a cluster. That is, it's in the neighborhood of a core point. And finally, an outlier point not be by a cluster. A point Y is said to be reachable from X if there is a path P1 to Pn, with P1 equals X and Pn equals Y, where each Pi plus 1 on the path must be core points, with the possible exception of Pn. An object Y is directly density reachable from object X if X is a core point and Y is in X's epsilon neighborhood. A is directly density reachable from B. B is directly density reachable from C. A is indirectly density reachable from D. D is not density reachable from A, since A is not a core point. Density-based clustering works by taking a random point that does not belong to a cluster or is an outlier and determines if that point is a core point. If not, label it as an outlier. Now we'll pick another core point at random and determine if it is a core point. If it is a core point, then all the directly reachable nodes are assigned to the cluster. Then do neighbor jumps to all reachable points and add them to the cluster. Keep finding neighbors until you're unable to. This point would be labeled as a border. Now keep repeating steps 1 and 2 until all points have been labeled or assigned to a cluster. In this case, the last 2 points are also outliers. Now we have our cluster. At this point, it's important to know the difference between density-based clustering, or DB-scan, and k-means. DB-scan is effective at cutting the noise out of data set by labeling them as outliers, whereas k-means uses the entire data set. In this example, we have 2 sets of clusters resembling half moons, each belonging to different cluster sets. Now, if we add some noise, we can see if DB directly assigns the cluster. Let's run DB-scan.
we'll see that it'll correctly group the top half group as a cluster, and it'll do the same for the bottom half. Then it'll predict the rest of the points are outlier or noise, which is correct for it. Now, let's look at the same half-moon example using k-means. This will also include the noise data points. Now, two random centroids start off, and the collective points are assigned. Then, each centroid is moved accordingly to their new location. And once again, the collective points are assigned, and the cycle repeats until the centroids are at the point where no new points are assigned to either side from the movement. Once k-means is complete, we can see that it did not correctly predict the half-moon clusters, nor the noise. K-means has a huge disadvantage in that it considers the whole data set and cannot distinguish noise from cluster grouping. It might also identify clusters incorrectly due to its reliance on centroid placement.
Any other questions? All right, I think I already covered everything supposed to be here. And I will see you Thursday in computer lab. And we will test out some of these algorithms. Okay, good.